So let's talk about mutations. This is what the syllabus objectives say. There's several, so I'm going to put these up again at the end so you can go through and check that you have covered them all off. I'm also going to put up the guidance, the syllabus guidance that goes with these syllabus objectives, just to make sure that you understand what you don't need to know. And now we're talking about chromosome mutations. So when it's a mutation of a gene, it only affects the expression of that one gene. But remember, chromosomes might have thousands of genes. So if you have a mutation of a whole chromosome, then it's going to potentially affect all of the genes on that chromosome. And that's why we talk about chromosomal disorders causing syndromes. A syndrome is a condition that's got a whole wide range of issues affecting large different parts of the body. We're talking about things like Down syndrome, and I'm going to give you some more information about that one soon, but that's a chromosomal disorder, and you probably know that Down syndrome, people with Down syndrome actually have a whole raft of different signs and symptoms of that condition associated with the mutation. So more on that soon. Chromosomal damage or changes to the chromosomes can affect the structure of one or more chromosomes, and it can affect the number of chromosomes. Firstly, about the structure. So if there's errors in meiosis or damaging agents, so remember, meiosis is the process of producing the gametes. So if you have errors in meiosis or exposure to damaging agents, it can actually cause breakage of a chromosome. And there's several different ways that this can occur. Deletion of a segment, duplication of a segment. So we're talking about a segment of a chromosome. So, maybe a large part of a chromosome is actually missing, or it's duplicated, or it's inverted, so it's upside down. Or we have translocation, and that's what this picture is here. So there's been a swap here between segments of the chromosome. So in this case, nothing's missing, but it's in the wrong spot. So, that's going to be an issue for gene expression as well, potentially. All right, so that's changed to the structure of chromosomes, but we want to talk more about change to the number of chromosomes. One of the ways that you're required in terms of the syllabus to be able to identify these chromosomal disorders is by looking at a karyotype. So what's a karyotype? It's the appearance of the complete set of chromosomes in the cells of a species. So they're presented like this, and as you know, with the human genome or the human chromosomes, we have 23 pairs of chromosomes, 22 pairs of somatic chromosomes, and 1 pair of sex chromosomes. And of course, for female, human who's assigned female at birth has 2 X chromosomes, and somebody assigned male at birth has an X and a Y chromosome. So we can look at a karyotype and identify whether there's any differences to the normal number of chromosomes. And there is actually a difference on this particular karyotype here. I'm wondering if you can find it. We're going to look at it again in a minute. Okay, so how do we get a change in the chromosome number? It's called non-disjunction, and it occurs during meiosis. Okay, so let's go back to our meiosis fingers. So we've got, remember how we have this, in this particular scenario, this organism has got 1 pair of homologous chromosomes. Okay, then we have DNA replication. So now we've got the tetrad, okay, or the 4 chromatids. And then in meiosis 1, we have the homologs that separate. And in meiosis 2, we have the chromatids that separate. Now let's think about a situation where we have non-disjunction. So in meiosis 1, how about these ones, these 3, don't separate? So we end up with this situation here at the end of meiosis 1. Now, in meiosis 2, what's going to happen there is that we're going to have this separation probably. So you can see that we've got this, this gamete here has a normal single copy of the chromosome, and this one here we have a single copy of the chromosome. This one here, we've actually got 2 copies of the chromosome, and this gamete over here, we don't have a chromosome at all. Okay, so that is potentially what could happen. So let's go through it again. So DNA replication, we've got our tetrad. Meiosis 1, this time we've got, well, let's say meiosis 1 occurs properly, but this time we have non-disjunction at meiosis 2, and so we've got this situation. So these two are going to be normal. This one is going to have 2 chromosomes, and this one here, no chromosomes. So that's what we mean by non-disjunction. The chromosomes, homologous chromosomes, or the chromatids, don't separate normally during meiosis. As a result, one gamete receives 2 of the same chromosome, and another gamete receives no copy. And we're running through the different scenarios here. We've got 2 homologous chromosomes, not just 1. Okay, but same deal. Okay, so there's some terminology. It looks a bit scary. There's a lot on this page, but it's not too bad. So, first of all, aneuploidy is any deviation from the usual chromosome number. Now, we've actually seen ploid before with haploid and diploid. Okay, remember, so we're talking about all of the chromosomes. Haploid was half the number of chromosomes, and that's in the gametes. And diploid is where we've got the somatic cells, where we've got 2 homologous chromosomes for each of the chromosomes. Okay, so we've already talked about ploid. So aneuploidy is where we have a deviation from the usual chromosome number, where it affects the somatic. So somal means somatic. When it affects those, 1 to 22 non-sex chromosomes, we call it autosomal. And if it involves the X or Y chromosome, the sex chromosomes, we call it sexual aneuploidy. Okay, now, so we've also got this term polyploidy. So poly many, ploidy meaning chromosome. So it means more than 2 complete chromosome sets. Okay, now a little bit more. You might have heard of trisomy before. So when we're talking about somy, so chromosome, so that's where some comes from. So more than 2 copies of a particular chromosome, we call it polysomy. So monosomy, when we only have 1 copy. So we're talking about in the diploid cells, but we've only actually got 1 copy of it. So that would be like in meiosis, we had a non-disjunction. So we have 1 gamete that's got 2, and 1 gamete that's got none. You had fertilization of the gamete that had 2 chromosomes with 1 from the reproductive partner that had 1. So then you would have trisomy. And if you had the one that had no chromosome with the reproductive partner that had 1, well, we're going to have monosomy. Okay, so hopefully that was okay for you. All right, let's talk about a couple of little examples then. So I mentioned that syndromes tend to have a wide range of signs and symptoms. So this is Down syndrome, and it's trisomy 21. So 21 tells us it's the 21st chromosome, and trisomy means that there's 3. And if you have a look down here, trisomy 21, we can see that there's 3 chromosomes. So there's a wide range of impairments and differences that are expressed as different signs and symptoms, and these would be all related to the genes on chromosome 21. Okay, now, so that's one. So let's have a look at another one. So one thing that you need to be able to do, this is from the syllabus, it says you need to be able to use a human karyotype to identify ploidy changes. Okay, ploidy changes and predict a genetic disorder from given data. Okay, so have a look at this karyotype and see if you can identify what the condition would be. So, first of all, is this male or female? You can see this person is female. And hopefully you can see that this is a trisomy. So this one's called trisomy 18, otherwise known as Edwards syndrome. Have a look at this one, see if you can identify the issue here. So this is non-disjunction of sex chromosomes, so you can see that there's two X's. So this individual is XXY, but so they've got two X chromosomes and a Y chromosome. This is called Klinefelter syndrome. And another one, so this one here is monosomy X. This is also called Turner syndrome. And so it produces females that are XO and so, or X0. So there's only one X chromosome and there's no Y chromosome. Okay, so whilst the Klinefelter syndrome individuals are assigned male at birth, the monosomy X or Turner syndrome, they're assigned female at birth because they have the X0